I've been very excited over the last 6 or 7 years actually about single-cell genomics, and I'll mention the human cell atlas a little bit at the end of my talk, but basically the bulk of my talk is going to be about the evolution of innate immunity. And this is actually a system that exists in all the cells in our body where we have an innate immune response to bacteria, to viruses, and it's a system that's very old in evolution, and we've looked at it in primates and rodents. Now, the reason that single cells are important is partly, obviously, as Orly said, because tissues are complex and we haven't actually discovered all our cell types. But another reason is actually that even within a fairly homogeneous population of cells like fibroblasts, which is the system I'll be looking at, there may be cell-to-cell variation in the resting population and even during the dynamics of a stimulation. And that, in this beautiful, colorful picture, we see the differences in protein abundance reflected in different fluorescence levels of cells. And of course, imaging has been used as a single-cell biological technique for sort of decades, fluorescence microscopy. And we can now sort of take a step forwards in terms of understanding cell-to-cell variation using single-cell genomics, and single-cell RNA sequencing specifically is what I'll be talking about. So the cell autonomous innate immune program is a program that essentially exists in every cell, and the antiviral element of that goes through viral receptors in our cells, many of which sense double-stranded RNA, and then signal through transcription factors to kick off a program that sends other cytokines outside of the cell, which leads to a positive feedback and a secondary response sort of to the neighboring cells. And these cytokines signal not only to the cells in the tissue, and cell death is one element of that, they also signal to the adaptive immune system and can kick off a sort of cytokine storm. And the single cell element here comes in because of the speed at which individual cells signal and the pathways that they can go down are quite heterogeneous. So there are metabolic changes that can take place. Cell death is an important one that leads to tissue damage, and then inflammation interacting with the adaptive immune system, and so on. And at the molecular level, the interferon-stimulated genes that come through this type 1 interferon signaling pathway range from cytokines, so other secreted factors, through to transcription factors and enzymes that are involved in these processes. Actually, I've got a forward. Now, I want to, actually, I'll step over here so that you can see this. So from an evolutionary point of view, as I said, this pathway is present, sort of is essentially there throughout vertebrates and invertebrates. But it's, as you may expect, sort of there's an interaction with the pathogens that you can see would lead to a rapid divergence in detail of the individual factors because we're in a constant arms race with these pathogens. And so that leads to many of the factors involved in these pathways are actually amongst the most rapidly diverging genes. At the same time, there is this arms race with viruses, and the pathway, needs to be strong enough and speedy enough to cope with all the, an average of 6 or 7 viruses that we're dealing with at any one time in our body. But at the same time, if you have an overactive immune system, you can see that you would get this cell death problem and tissue damage. And so there's a balance there that's happening in evolution between the arms race with the pathogens and the need to sort of, at the same time, kind of avoid tissue damage and dampen the response. And from a genetics point of view, many of the factors that are regulators of the pathway specifically have been identified as being mutated in disorders with abnormal upregulation of type 1 interferon. They've been dubbed interferonopathies by Yannick Crow in a recent review. And what interferonopathy means is that in these diseases, there's a ton of interferons in your blood of these unfortunate people, and of course that we know mechanistically that must be signaling through the innate immune response and lead to cell death in tissues, and this is kind of the hallmark of many of these diseases that are manifested in various tissues, and also potentially to the adaptive immune system, T cells, B cells, and so on, to stimulate them. And so here are some of the examples: systemic lupus erythematosus, Aicardi-Gouttiere syndrome, and so on. So it's really quite a ubiquitous, an important central pathway. And the question that we're addressing here is, what's the evolutionary architecture of the innate immune system if we look across organisms? And how do these constraints to regulate the system and prevent it from being overactive, how do they play out in contrast to the pressure to respond to pathogens and to be in this arms race with the viruses? And how do we get tight regulation despite this rapid evolution, and also the cell-to-cell variability that I mentioned before that we see in this system because of divergent cell fates, essentially. Okay, so the way we addressed this was by taking primary dermal fibroblasts, so basically skin cells from the shoulders of female humans, macaques, rats, and mice, so primates and rodents, and then stimulate them with poly-IC, with double-stranded RNA, which is as I said, many of the sensors of this pathway for viruses go through double-stranded RNA binding. And then track the response over, it's a fairly rapid response that happens in the cells over 8 hours, roughly or so, by both bulk transcriptomics and also single-cell RNA sequencing to get a measure of cell-to-cell variability, so the heterogeneity in the response of this system. And then we also used chip sequencing for chromatin marks and sort of looking at the hardwired features of the genome in terms of sequence evolution, promoter evolution. So this allows us to trace the cross-species transcriptional changes over this 8-hour response to double-stranded RNA. And you can see the kinds of different patterns illustrated very simply here, where we've got humans and macaques, and then the rodents. And there are some genes that basically let's say if we just look over the total fold chains over 8 hours for the sake of simplicity, then we have some that have a conserved response, similar across all organisms, some that are rodent or primate specific, and some that have a response that's species specific. So we call this response, the response divergence, if there's an evolutionary change in the gene expression pattern. So the genes are there in all the organisms, but they're responding in different ways to this viral mimic. And the way we can quantify this is using a model. We take the genes that are differentially expressed across the time course and then home in on the ones that are one-to-one orthologs and quantify whether they're responding in the same way, sort of lowly divergent in terms of their dynamics of response, sort of medium level of divergence, or whether they are very different across the species. And we can, as I said, response divergence here basically means the genes are present, orthologs are present, but the way they're responding to the viral mimic is different across the organism. So there's an evolution of the gene expression pattern. And we can model the extent of divergence using this Ornstein-Uhlenbeck model with Brownian drift and then a selection force. This was originally introduced by Daniel Hartle and has been sort of refined over the years by different people. And so the questions that we're asking of this data is, what's the evolutionary architecture of innate immunity? What's the pattern in terms of conserved expression and divergent expression? How do these regulatory constraints balance against pathogen pressure and, as I said, the cell-to-cell variability? And so if we start off with looking at the global sequence features sort of, and gene evolution relative to these different levels of response divergence, that we take the lower quartile, the most conserved genes, the medium 50%, and then the highly divergent genes, and then look at DNDS, sequence evolution across these groups, what we see is that there's a very clear and significant trend for the highly divergent genes in sequence to also be the most divergent in expression. This is a kind of monotonic increase, essentially, continuum in terms of sequence divergence and expression divergence. independently of sequence evolution, we can sort of look at conditional probabilities and find that the families that are gained and lost at the highest rates are also more divergent. Gene age, so when these genes popped up in evolution, is more recent for the highly divergent genes. And the number of protein-protein interactions, so the constraint from physical interactions inside the cell, is highest for the genes that are most conserved in expression, that have the most conserved expression across the organisms. So basically there's a kind of parallel picture going on here in terms of sequence evolution, gene gain and loss, and gene age, and the expression patterns of these one-to-one orthologs across rodents and primates. So it's telling us that in innate immunity, transcriptionally diverging genes are also diverging fast in sequence. There's fast gene gain and loss, they have a young age, and they have few protein-protein interactions. And all of those things together may appear to be telling us that there's basically a relaxation of constraint for this class of genes. So there's an absence of selection pressure. Now, the kind of surprising thing about this is that I'll come on to a little bit later, is that many of the most diverging genes are cytokines, chemokines, and so on that we know are functionally important and that are actually driving this response, the interferon itself, interferon receptor, and these genes. So there's a kind of cognitive dissonance, or there's a mystery there, a question kind of at the heart of this. These things are diverging very, very quickly, and yet they're so functionally important. I'll come on to that. But the global picture is that we see that things that are diverging in terms of the response, the transcriptional response over this 8-hour fast, rapid innate immunity, are also divergent in sequence, and they aren't involved in a lot of protein-protein interactions on the host side in the cell. Okay, now let's try to understand this and dig into this a bit more mechanistically in terms of the regulatory constraints and the pathogen pressure. Well, if we look at the interactions, now I was talking about protein interactions in the host, in the human and mouse and so on. Now, what about if we look at host virus interactions? Then what we see is that there's again a kind of continuous pattern in terms of the number of interactions with viruses, where the fastest expression divergence kind of gene categories have the fewest viral interactions. And genes that do have protein-protein interactions with the virus and that are also targeted by immunomodulatory factors, viral factors that are known, that have been described in the literature to modulate the immune system, they are actually more conserved. So again, here the genes that are diverging in expression have no interactions with viruses, and the genes that are more conserved across organisms are the ones that are more constrained by viral interactions. So there's a consistent pattern here that the viruses are targeting conserved innate immune genes, and the genes can kind of escape and get away are the ones that are more divergent, which it makes sense, but it's all kind of, it plays out in this quantitative way in terms of expression as well as in terms of sequence. And if we look in detail at the genes that have viral interactions, then it's the number of interactions is also proportional to essentially the extent of sequence of expression divergence, transcriptional response divergence to double-stranded RNA. So again, the ones that have a larger number of interactions have more conserved expression and medium and high, and it's a kind of continuous decrease in terms of the number of host virus interactions. So these genes that are diverging in expression across the species, in this cross-species comparison, in human have the highest number of host virus interactions. So pathogens target genes that are conserved in expression and in sequence, and the genes that can sort of get away, that can escape, are the ones that are diverging both in sequence and in their cross-species expression pattern. Now, the second half of this question is, yes, we have this pathogen pressure for rapid evolution, but I also told you that if we have a massive antiviral response, we risk basically having tissue damage and autoimmunity. And so how are regulatory constraints sort of balanced against this need to escape? And so if we now look, I've talked very broadly in very global terms about different speeds of divergence. But now let's try to understand functionally what these different categories are. And so if we break down into their molecular functions and annotations, basically the different categories that I described that are part of the innate immune response in the first slides, then we have the viral sensors that are actually sensing the double-stranded RNA, for instance, cytokines and chemokines, which are these proteins that are spewed out by the cell and are involved in signaling, the receptors, and then signaling factors, ligases, deubiquitinases, and then regulators like transcription factors, chromatin factors, and signaling enzymes and other enzymes. And what we see is that the most divergent categories are the sensors, cytokines, and chemokines, and the receptors. Basically, this group isn't significant, but it's these three groups. So it's the signaling factors and their receptors. And the ones that are most conserved in expression are the ligases, the regulators, and the enzymes are more conserved. And this is sort of the average across all the genes, and you can see these are above average and these are sort of below average. And how can we understand this? Well, one way of understanding it is in terms of this host pathogen race. That's one sort of framework. Another one that I think is interesting is this sort of bow tie architecture concept put forward by Oriolon that explains a lot of sort of evolutionary patterns. And I think we can also apply it here in the sense that cytokines and their receptors and the viral sensors and the receptors are sort of the input of the system, if you will. And the bow tie here means that you've got sort of rapid evolution or diverse sequence space at the top of the bow tie. The middle layer is more constrained, and these are the enzymes and the regulators. And then the output are the signaling molecules here in a way. So the viruses are targeting these guys and constraining them, and this is also the middle of the signaling pathway, whereas these guys and these factors are basically diverging quickly and can sort of get away from the viruses and are basically balancing this sort of balancing act between pathogen pressure and yet the need for having a controlled regulation. So if we look at, as I said, if we look at the sequence conservation, then there's a coherence basically between expression divergence and sequence divergence by DNDS, or coding sequence divergence. What if we look at regulation in the promoter, conservation upstream and non-coding sequence in the promoters of these genes, and we take the transcription start sites and then go backwards about 500 base pairs and look at, using phylo-P, kind of quantify how conserved this promoter region is. A really interesting pattern emerges, which is that the genes that are highly divergent in this response, the cytokines, chemokines, and their receptors, and that are also the ones that are actually divergent in their amino acid sequence, these are actually most conserved in the sequence of the promoters. So the transcriptional regulatory region that you might think might be really important for these genes is actually most conserved in sequence. And the most conserved in expression, these light blue ones here, are actually the least constrained in terms of their promoter sequence. And now if we try to understand this and dig into what's known about promoter architecture, this review from Boris Lenhardt is actually really good at summarizing this, and he's worked on promoters for many years. And of course we know that there are TATA promoters that are devoid of CpG islands and that have this motif that binds the RNA polymerase, TBP, in a very specific and strong way. And then the other class of promoter is the type 2, or sort of more ubiquitous promoter, which is a CpG island promoter and is TATA depleted. And so if we now try to understand this conservation pattern in terms of these different classes of promoters, we can see that the TATA promoters are enriched in the conserved promoter architecture, which are the more sequence divergent and expression divergent genes. And the CpG island promoters, which are TATA-less, they have these broad CpG islands, as you know, and the RNA polymerase can start in a kind of staggered way, kind of with a non-specific binding. They're the genes that are actually more conserved in terms of their expression pattern and in terms of their sequence. And so we have basically this picture where we have conserved promoters that belong to the Tata CGI-less class, but the genes are actually highly divergent in expression, and the amino acid sequences are divergent. On the other hand, we've got genes that have the CpG island type promoter, which is there's less conservation than we have in these Tata promoters, which could also be to do with transcription factor binding site density, and these genes are actually less conserved in terms of their promoter, but more conserved in terms of their cross-species expression pattern, their amino acid sequence. So there's something very interesting about this promoter architecture that allows the divergent genes or that may mechanistically be linked to their expression divergence. And this, of course, as I said, these are cytokines, these are enriched in cytokines, chemokines, and their receptors, this most important class of signaling factors in both innate and adaptive immunity. And this promoter class, you can see, because of this very specific Tata motif, is conserved, but is leading to a divergent expression, and is presumably allowing these genes to also sort of escape from the viral response. So Tata, I'm not sure how many people are aware, but basically the Tata binding site through this strong affinity to the RNA polymerase also means that the K-on, K-off rate of the RNA polymerase is basically slower than for the CpG island type promoter, where the RNA polymerase can actually bind at many sites and have a sort of high frequency of binding, leading to a high frequency of transcriptional bursting. And so what that means is that the transcriptional sort of bursting pattern of Tata promoters has been shown to be more stochastic mostly in yeast, work from Nama Barkai, Aaron Segal, and so on. So that basically brings us on to how cell-to-cell variability is basically regulated in this system, and how you can get a tight regulation despite this evolutionary divergence in cell-to-cell variability. So how do we measure cell-to-cell variability? Well, we come back to my favorite technology, which is single-cell RNA sequencing. And what this allows us to quantify is the variation in a particular, in a cell culture, in a cell population of a given organism, any of the four species that we're looking at. Here we're using human cell-to-cell variability quantified using the distance to the median for each gene in the population. I won't go into the methods, but basically it's the breadth of the distribution of the expression of that gene in the population. And so if you have a broad distribution, basically there's a high cell-to-cell variability. If you have a tight distribution, then you've got a very tight regulation of the expression of that gene. And what we see, which is quite astonishing, is that basically for the more conserved genes, there's less cell-to-cell variability, and for the higher genes, there's more cell-to-cell variability. And this is independent of expression level. We're normalizing for that. It's independent of the speed of the response, because we can sort of look at this feature over time, and it's conserved sort of regardless of which species we're looking at. Now, again, when we relate cell-to-cell variability to the promoter architecture, I've already told you that there's some sort of molecular biological studies on the mode of action of these promoters, and there's also genomics data from yeast that suggests that Tata promoters would have a higher cell-to-cell variability, a lower burst frequency, essentially, of gene expression. And then and that's exactly what we see in this mammalian system, where this hasn't been shown before. And so we show basically, I think for the first time in mammals, that cell-to-cell variability is linked to this Tata promoters, and the CpG island promoters here in blue have lower cell-to-cell variability. So it's telling us that highly divergent genes across species also have high cell-to-cell variability within species. And at a molecular level, this is the category of Tata promoters and CpG-less. And the conserved genes, in terms of expression across species, also have low cell-to-cell variability within a species. So there's got more homogeneous expression in terms of their transcriptional kinetics within human, within mouse. They've got these CpG island promoters, and the cross-species expression pattern in this innate immune response is conserved. So that really so it brings me to the conclusion that we see the highly divergent genes in terms of transcription also have high cell-to-cell variability within a given species. We can link this mechanistically to the different promoter classes, and to kind of sort of bring this together, what it sort of philosophically, what it's indicating is that there's an evolutionary adaptability that's linked to high cell-to-cell variation. Sort of heterogeneity in the response within an organism is also linked to evolvability essentially across organisms. Kind of, that's the sort of big picture. How is that possible? Well, as I mentioned, there's literature in yeast kind of that's many years old now that links nucleosome architectures and promoters within species to also evolvability across species, as it were. And now, I'm not saying that it's possible to actually evolve sort of adaptability, but it seems that in yeast, these are stress-related genes. Now we're seeing in mammals, these are innate immune response genes, and it is possible that basically the response, needing to respond quickly to a change in environment, needing to respond quickly to these viruses, is also is linked through the molecular kind of organization of the Tata promoters also to adaptability in evolution. And if we dig in, just to make this a bit more concrete for you, we've got here different molecular classes. In pink, again, the cytokines and chemokines. In green, the transcription factors. And light green here, the enzymes. And if we look at this in 2 dimensions, cell-to-cell variability and divergence across species, you can see these guys, basically the top right-hand corner, high cell-to-cell variability and high cross-species divergence in expression. And this is conserved basically across the time course, as I mentioned, and it's conserved across species. It's a general pattern. And so at a less philosophical level, a more concrete level, it's the cytokines and chemokines and their receptors that are diverging rapidly and are highly variable. It's conserved across conditions and species, and we can link it to this promoter architecture. Now, you could say, yeah, it's absence of selection. We don't think that that's the explanation, because we can see, as I said, there's this particular promoter architecture, and there's also co-expression patterns that we can extract the single cell level between the cytokines and chemokines and upstream regulators. And we can do this network reconstruction from single cell data. It's one of the beautiful features of this high-dimensional complex data sets, that you can then use gene-gene correlations in a very powerful way and basically reconstruct the upstream regulators with the cytokines. We see them co-expressed in particular subpopulations of single cells in the data. And many of these upstream regulators, that you see the cytokines in purple, CXCL10, interferon beta, IL-6, and so on, are co-expressed in single cells with their upstream regulators. And these are enriched in the molecules that I told you about earlier, which are where the mutations lead to interferonopathies. So the story that I've told you is basically a sort of dissection of the evolutionary architecture of this ubiquitous innate immune response, ubiquitous in our body and ubiquitous kind of across the evolution of animals. And what we see is that genes that are divergent in their cross-species expression are also divergent in the coding sequence. The pathogens are actually targeting, kind of counterintuitively, they're targeting the conserved genes, and the genes that are divergent in expression and sequence and escaping these pathogens actually have these conserved Tata promoters. And those are the genes that have high cell-to-cell variability. And these are the genes that we also believe, despite their divergence, they actually have regulatory and strong functional importance. So there's a contradiction, there's a kind of contradiction there. This is a sort of visual summary. You've got high divergence in response, low divergence in response, high cell-to-cell variability for this class of genes, low cell-to-cell variability for these genes. Coding sequence evolution contradicts promoter sequence evolution in both cases, and the viral interactions are enriched in the more conserved genes. The star of this show actually is someone I haven't mentioned so far. It's Tsachi Hagai, who's an EMBO and HFSP fellow in my group, and he's been absolutely brilliant in, or at least at my lab, does both wet and dry work. And actually, a lot of the time, this is split between two different types of, or two different categories of people, as it were, in the group. In this case, it's actually unified in this person. So he did both, lots of the whole wet work, essentially, and the whole dry work. So it's a real tour de force by one brilliant postdoc. He rallied many of the other group members to help him with bits and pieces, and we've had fantastic collaborators on the human genetics with Gosha, and making sure that we're doing the right thing in terms of our interpretations of data, and donations of samples from places in the UK and the Netherlands. I just want to say the outlook for the future, and as head of cellular genetics, something that I'm very keen on is human genetics at the cellular level. And where this project is going in its next incarnation, its next phase, is that we're not just looking across species, but also across human individuals. And so we're taking dermal fibroblasts from many Cambridgeshire individuals who've donated their shoulder skin, and then stimulated them and done single-cell RNA sequencing of this 6 to 8 hour response, where we can then reconstruct the genetic variation that's linked to cell-to-cell variability in the response. And that's something we're very excited about in the future. You've heard a little bit here about how powerful single-cell RNA sequencing can be. And it's really been an amazing journey over the past 8 years or so in this world of single-cell genomics. I think it will impact this community, which I'm really delighted to be invited to be part of in a very big way soon. And for me, what the kind of revolution, the resolution revolution in genomics, and the scale and low cost of these technologies, what it's meant is that I think that the time is now there that we can actually dissect the entire entire organisms at the single cell level using a combination of single cell RNA sequencing and spatial mapping. And cells are the units of life and of gene regulation, as we've just heard, but we don't actually, there are many aspects of cell state, cell type, and so on that we haven't resolved comprehensively, surprisingly. And so the idea of the human cell atlas is that we essentially make a sort of Google Maps for the human body, that we have, on the one hand, very high resolution using single-cell RNA sequencing, and that we also have coordinates for the body using spatial methods like spatial transcriptomics, imaging methods, and so on. We combine these two things and get a Google Maps where we can sort of zoom in and out into tissues and organs. And if you're curious about this project, then please visit the webpage, please talk to me at the conference and the breaks and so on. And we have regular meetings. It's a growing community involving computational people. There's a genetics element to this that's very important. Clinical community, technology developers, and so on. It's a real pleasure to actually see this evolve and grow and push this forward with Aviv Regev, who's my co-chair for the organizing committee. I also want to make a shout out for the Single Cell Biology Conference here in March, if you're interested in coming back and you're excited about the power of single cell biology. And I'd also like to enlist your help in advertising a faculty position, so the first search that I chair here for the cellular genetics program at Sanger, where we're looking for faculty in a diverse set of areas, some of which may be relevant to you, or tell your friends and colleagues, please. And thank you for your attention. I'll take any questions.
I had this question about, you showed that these highly divergent genes with highly divergent expression have conserved promoters with data bugs. Do you know if these genes or these that the promoters are more likely to be regulated by distal enhancers, and you would maybe see variation in the enhancer sequence and not the promoter sequence.
Quite possibly, yeah. Yeah, I mean, the regulation is complex. It's obviously not all in the promoter, so yeah. I mean, I don't see an easy way of accessing that, but it's, yeah. I mean, as I said, there's also, there's a kind of more simple explanation, which is basically that you have a small motif that's responsible for the regulation, and so that's easier to break or change, I would say, than a large CPG island where you've got to change lots of things to actually impact this much more robust architecture of transcriptional regulation where the RNA polymerase can kind of bind at many places. Yeah, but it's going to be a combination of factors. Dan? Yeah.
Hi, Sarah.
Dan
Gaffney from Sanger. So I had a question about, can you give us a sense of what's known about disease at the level of single cell? I mean, you talked about this variability in response. I think we're often thinking about loss of function mutations and so on, but are there well-known cases, or are we capturing cases where disease mutations lead to loss of heterogeneity? Like heterogeneity is the sort of healthy condition, and the disease mutation leads to absolutely every cell responds.
Yeah, that's a great point. Yeah,
but
that's
do those things exist?
That's basically what's happening in many of these diseases. This pathway is constantly. So instead of having this kind of healthy balance of stochastic expression of the cytokines, the cells are all in this overactive state where they're chucking out lots of interferon, and you're getting more homogeneity in that sense, which is pathogenic. Yeah. I think there's a question here, Orly.
Eleanor Schiff from UCL. So you spoke about the divergence. You said you don't think it's because of a lack of selection. So how did you show that, or why do you think that?
Well, basically because, as I said, the cytokines and chemokines and the receptors are so functionally important, A. B, you're balancing against, you've got conserved promoter architecture. And I mean, that's the main explanation, really. And C, because you've got this, in this particular scenario, you've got this continual need for escaping the interactions with the viruses, and these are the proteins that are devoid of viral interaction. So the host pathogen arms race basically is going on at the same time. And we see this, the conserved genes are targeted by the viruses kind of more frequently. Does that make sense?
I can speak a lot. Vijay Sankar from Boston. So with the interferons, you can get differences in levels of the cytokines and receptors that mediate variation in cellular responses, like antiviral responses versus anti-proliferative responses. And I was wondering to what extent you also think heterogeneity at that level could, and I don't know if you've looked at that, in terms of their ability to mediate anti-proliferative versus antiviral
So the
responses.
cell kind of, absolutely. So the stalling of the cell cycle and then going down the sort of pathway of cell death, that's one of the main things I think that's sort of balancing against the antiviral response, right? Because if you go down the apoptosis pathway, then if there's too much of that, you basically lead to this pathogenic tissue damage and these autoimmune diseases. Now, so we haven't studied those mutations kind of specifically, but sort of that's the balance that you've got going on there. Yeah. I mean, I'm happy to discuss more if you have a specific idea about levels of cytokines. Yeah.
Casper Leitch, also from Boston. I was just curious about the protein interaction experiments that you described, which technique you used, how you quantified.
They are published data sets.
Oh, these are published data
sets. Yeah, these are all published data sets. Yeah.
Have you considered generating them de novo for the right cell type, right perturbation state?
Yeah, it would be interesting to dig into particular protein-protein interactions in more detail, but that's kind of not in the scope of this work. Yeah.
Sure.
So the data that we generated, I mentioned, or maybe it wasn't clear, I mentioned it on the first slide as an overview. It's the genomics rather than the proteomics. So the expression at the bulk level, the single cell level, and we actually also use chip sequencing that I didn't talk about for the promoter. If I go back to that slide, it was here somewhere. Oh yeah. Go away. Yeah, and the protein interaction data sets and so on are integrated from the literature. I think there's a question here, Orly.
Okay, I'll go with that one. So I'm Nathan Skeen from the Karolinska. So I was wondering if you'd looked into whether... Hi. I was wondering if you had considered whether the genes which are more divergent in expression, are they enriched for heritability for autoimmune disorders? Or is there any...
Their regulators are.
Mm-hmm.
Okay. So, sorry, maybe it wasn't clear. So you can see on this slide is kind of summarizing this review, what's known. You can see here are the factors that are known to be involved in these diseases. And then if we do this correlation analysis, what we see is that the cytokines and chemokines that I mentioned are correlating and are co-expressed and controlled by these genes where you have the mutations that lead to interferonopathies. So it's not the cytokines and chemokines themselves, but it's their regulators that where the mutations, we think the mutations are happening.
Robin Walters, Oxford. How many separate genetically independent cell lines were your results based on for each of your four model species? So it's 3 to 6 for each species. Sorry?
Yeah. Between 3 and 6, yeah.
Could any of the variability you see between species actually be sampling, to some extent, individual level variability in regulation?
I mean, potentially, I would say unlikely for the humans and the primates because they're very genetically diverse, kind of. The mouse is an inbred mouse line, and as is the rat. But we do have for the primates and the human, which is of particular interest, and then the rodents are further away anyway. I don't think it's a problem. And actually, for the humans, we've got, as I said, we're collecting this data set of 70 individuals. So yeah, I would say no.
Hi, Sarah Brown from Dundee. Thanks for your talk. It's really cool data. I was wondering why you chose dermal fibroblasts as the cell that you've focused on for all these studies, especially with your interest in autoimmune disease.
Yeah, so good question. So I come from an adaptive immune kind of, T cells are actually my favorite cell, so why do we use fibroblasts? So it turns out that for viral infections, the fibroblasts are actually highly relevant and a standard system for the antiviral assays. And although we think that a lot of the autoimmune diseases are linked to the adaptive immune system, and many of the associations are, there are actually a lot of associations also with this innate immune response and with the tissue. So the structural cells in the tissues, like the fibroblasts. So actually, I kind of had the same question in my mind that you had originally. But then, actually, if you dig into I showed these type 1 interferon regulators. They're all in this innate pathway. It's not a T or B cell. They're not T or B cell relevant genes. And if you look at
the diseases don't particularly.
So I mean, the other reason is basically that in terms of in terms of the previous question, the genetic variability, we have here the Hipsky resource, human-induced pluripotent stem cell, and the dermal fibroblasts are taken from the same donors, where we have this huge genetic variability. I mean, there are 600 donors or something like that, and so we have that resource for these skin cells. We don't have it for cells of the adaptive immune system. And there are many other problems associated with that because you have with the adaptive immune cells. because there's more of the signature of the infection basically in those. But yeah. Good. Thank you.
So while we have you here, I was going to ask you, if you tell us a little bit more about your favorite method, single cell. What do you think has been the main challenges, technical challenges, in moving to look at gene expression in human tissues, and why was it that you wanted to found the Human Cell Atlas?
Mm-hmm. So I think the real motivating factor, so as I just mentioned, my background is actually, my favorite cell type are T cells. And it turns out that CD4 T cells, for instance, have this massive plasticity in cell state. And I realized that using bulk RNA sequencing, basically you're masking all of that, and it's kind of a, I wouldn't say pointless, but basically you're simply not drilling down to what is actually happening in the cells. And using flow cytometry or imaging methods, of course, you're limited to the number of colors that you can see at the same time, the number of markers in the cells. And so the single cell, in the early days, it was single cell qPCR, kind of in microfluidics that allowed you to go up to 48 or so genes in parallel. And then in 2009, we had this first paper where there was single cell transcriptomics. And that, for me, that was a kind of like sort of step change, basically, because I realized we could kind of go to this holy grail of actually understanding the expression pattern of single cells in a comprehensive way. And then when we started looking at the complexity of the CD4 T cell compartment, we discovered a steroid-producing T cell type. Many other people looked at, Azim Sourani was looking at primordial germ cells. Stan Linnerson started looking at, and there's obviously the whole tumor field and the neurons, and there were new cell states, new surprises that were popping up everywhere that this powerful high-resolution sort of microscope of genomics was being put. And then I realized that if you take this to the extreme, you're going to basically go to the whole body, right? And we can kind of do this human genome project for the body now. It's within reach. It's kind of becoming affordable because of this also scalability and technology development. Now, the one thing that we need, single cell genomics isn't kind of the sort of panacea, because what we also want to have is the knowledge about how these cells are positioned relative to each other in many tissues. So the immune system is kind of a special one, T and B cells, because they're very mobile. But for most tissues, and certainly for dermal fibroblasts and so on, you want to know how these cells, how is this innate immune response kind of radiating out between cells? How are the cells talking to each other? And so what we also need is spatial methods. And I think in terms of the genetics, the human genetics of this, there will be a time. So at the moment, we're basically at the point where we just want to have a human cell atlas, just one, like would be nice. Thank you very much, kind of in the next 5 or 10 years. But actually, for individual cell types in the long term, we'll have lots of cell atlases, and it will become a tool for genetics, I think, in the long term. Just like the human genome, we had a human genome, but then we had, we've got, what did you say, 14,000 or, whatever. 100,000 on the Wellcome Genome campus here, the 100,000 Genome Project. So that's really the kind of potted history, or sort of kind of the human cell atlas in a nutshell. And how I see single-cell genomics in a few sentences. Yeah.
Thank you very much. Thank you.